In human cells, there is a nucleus, and in the nucleus, there is a genome. Inside the genome lies 23 pairs of chromosomes. Forming the chromosome is a long strand of DNA. Within DNA, there are genes, and that is what you are seeing right now on your screen. Transcription is the first step in turning DNA into a protein. This process is where one strand of DNA is copied into a new molecule known as mRNA, or messenger RNA. Firstly, an enzyme known as RNA polymerase creates a complementary strand called the mRNA backbone. The backbone is made up of ribose sugar, phosphate, and nitrogenous bases. 3 bases in the nucleus attach themselves to the backbone depending on the DNA code. The purpose of this is to switch the bases with their complementary pairs. However, T turns into U in mRNA. Before the messenger RNA can leave the nucleus, RNA splicing must take place. Before splicing occurs, the mRNA is known as pre-mRNA. Then, however, introns, which are non-coding sequences of genes, basically junk DNA, are removed. Once this is complete, the mRNA is mature and is ready to go. Translation comes in 3 steps, the first being initiation. Before this can start, the mRNA moves out of the nucleus through nuclear pores and into the cytoplasm, and onto the rough ER, where it meets a ribosome. The ribosome is split into 2 subunits. In mRNA, the instructions for building a polypeptide are RNA nucleotides, A's, U's, C's, and G's, read in groups of 3. These groups of 3 are called codons. You can find what each combination is using an RNA codon chart. Each codon is met with an anticodon. An anticodon is part of a tRNA molecule attached to an amino acid. Once the codon and anticodon bind, it releases the amino acid. The start codon, usually the first amino acid to be produced, not always though, produces the amino acid methionine. Then, as every anticodon binds with its respective codon, the next amino acid attaches to the previous through a peptide bond. This then forms a polypeptide chain. The chain then molds itself into a 3D structure to form a protein. Once this process is complete, translation has reached termination. We now know how to make proteins, but how do they function? Insulin is an extremely important protein in the human body. Insulin is a hormone made by the pancreas that allows your body to use sugar from carbohydrates in the food that you eat for energy or to store glucose for future use. Insulin helps keep your blood sugar level from getting too high or too low. It is located on the short arm of chromosome 11 at position 15.5. Insulin has around 2 million base pairs. Its introns are at 0.2% versus 99.8% of exons. Environmental factors that can cause change in DNA sequence are known as mutagens. Mutagens can affect the amino acids and proteins produced in protein synthesis. An example of this is point mutation. Point mutation only has to change one of the nitrogenous bases. The effect of this could cause disease or even kill the organism. The most common form of monogenetic diabetes is also called maturity onset diabetes of the young syndrome. This is due to a mutation of 6 nucleotides out of 2 million. This shows how little change has to occur for you to have a disease for the rest of your life. Why do genes contain introns or junk DNA? A theory is that viruses in various points in evolution have placed exact copies of their DNA into the cell. The exact purpose of non-coding DNA is unknown. However, no harm has come to it being there, so scientists haven't had to do anything drastic to get it removed. Some scientists think it's not a bad thing, because all it would do is make the gene stronger and more diverse.